Good morning, everyone. Welcome to the next in the Viruses and Vaccines seminar series. My name is Helen Chu. I'm at the University of Washington, and today I am delighted to introduce our two speakers. First, we'll start with Dr. Trevor Bedford from the Fred Hutch, and he'll be giving us a quick update on SARS-CoV-2 genomics. And then our featured speaker today is Hanna Oltean, who is a senior epidemiologist at Washington State Department of Public Health. She will be giving a talk about genomic epidemiology and public health practice. She is also a PhD candidate at the University of Washington, and her work focuses on genomic epi, communicable disease surveillance systems, system design, implementation, and evaluation. So thank you, Trevor and Hanna, for coming to speak with us today. And I'll turn it over to you, Trevor.
Thanks so much, Helen. Yeah, just going to take at most 5 minutes here to walk over the latest with SARS-CoV-2. Just fixing Zoom a little bit. There we go. So as I think everyone is well aware, we've had this continual global evolution. Delta swept through in the course of basically a year. Omicron emerges, and then from October, emerging in October 2021, becoming dominant in January 2022, which is, I can't think of kind of a single thing in evolution that's been that fast at a global scale. Now we're getting these emergence of these kind of quote subvariants within Omicron that I believe should have their own Greek letters, but that's maybe a separate issue. So if we look in the US now, we've had kind of the original Omicron variant at BA1 that was dominant in January, February, March, coming back down, being replaced by BA2, then BA2.12.1, which had just a couple additional mutations on top of BA2, having its own kind of frequency peak, and more recently 4 and 5 kind of coming in, with 5 really being the one to become basically dominant, so that most infections in the US today are driven by BA5. We can break down case counts using this frequency data to kind of get an idea of how the epidemics have actually unfolded. We've seen a continually declining cases of BA1, BA2 here at kind of middling frequency, an obvious epidemic from BA2.12.1, and then BA5 still climbing, but looking like it's starting to get close to a peak at least, which is good. And then you can kind of see this in some detail here that we look at RT, so the number of secondary infections caused by one infection, and we see that, as you'd expect, that kind of new variants, when they emerge, have more secondary infections. As they cause epidemics, that creates immunity in the population, which drives down RT. So this is kind of what we'd expect from basically first principles here. And we're seeing kind of those same trend lines down for BA4, BA5. Better variants come in at a higher initial RT, cause larger epidemics, but as those epidemics come through, RT still declines. And you can see that I think this bump is probably artifactual, and it's really kind of getting close to hitting about 1. Then of course there's always the new thing. Right now the new thing, which should also have its own Greek letter, is BA.2.75 that's emerging from India. And so if we look at BA5, which had been kind of the current thing that's winning, it has these key mutations at 452R and 486V. If we look at 2.75, it has a good 8 mutations in spike protein, a number in the N-terminal domain, but also in around receptor binding as well. And if we just look at behavior, it looks like it's outcompeting BA5 in India. So it's pretty rare outside of India right now. Within India, we can see kind of data that's lagged by maybe 2 weeks, just as generally how it works, that we see BA5 kind of starting to be eclipsed by 2.75 here. And then if we estimate RT, we get an initial RT of over 2, which is again greater than the initial RT of BA5 of around 1.7, 1.6. However, there's this puzzle at the moment that case counts in India have not yet started to really peak, have really started to rise dramatically, as you'd expect from kind of this behavior. And so that's the only thing that's this doesn't look great. It looks like it could surpass BA5, but it's not clear what's going on with case counts at the moment. I think that should become clearer in about a week with more case count data and with more sequence data from India, and then we can maybe expect the UK as the kind of the next spot that we'd be able to have good data for 2.75 because it good travel connections to India and a really good genomic surveillance program. But that's yeah, I guess probably at least 3 or 4 weeks out probably to understand what's going on in the UK. And with that, I will stop and hand it over to Hanna to really discuss what's going on in Washington State, where we can look at these things in detail in Washington State because of her efforts and the efforts of Washington DOH. And I'll stop here.
All right, are you able to see my screen?
We can. You're not in slideshow mode. There you go. That's perfect. And for those of you on the call, you can go ahead and put your questions in the chat and we'll go through them at the end. Thank you.
Thank you all. Thank you, Helen, and thank you, Trevor, for the introduction. So my name is Hanna Oltean, and I'll be talking today about genomic epidemiology and public health practice and some of the history that's led us to where we are today in the field. So we're going to start all the way back with my very first encounter with genomic epidemiology and whole genome sequencing. And so this begins way back in July of 2010, when a 15-year-old boy was riding his ATV in South Central Washington. He crashed into an embankment and sustained knee wounds. Within 3 weeks, he had developed fever and cellulitis, and he was eventually diagnosed with coccidioidomycosis. This unusual case was reported to the Washington State Department of Health in September of that year. Later that year in November, the local health jurisdiction, as well as the Department of Health, went out to the field site and performed soil sampling where he had crashed. Specimens were sent to CDC, where they were then held for 3.5 years. We performed additional sampling in April of 2014, and results from this investigation were published in January of 2015. Whole genome sequencing at CDC was used to evaluate the relationship among 4 soil isolates, as well as the Washington 1 human case, and these were compared to 6 unrelated strains from California. No more than 3 SNP differences differentiated DNA from the Washington patient isolate and any of the Washington soil isolates, with a total of 10 SNPs found within this Washington clade. This is compared to more than 21,000 SNPs from the other regions represented here. The population structure of Coccidioides immitis is well known, and natural isolates are very diverse, with thousands of SNPs separating strains from the same geographic area. This near genomic identity that was discovered here in Washington indicates a recent common origin of at least these isolates. Coccidioides is an environmental fungus that persists in the soil of warm, arid regions with low rainfall. It grows as hyphae in the soil, which fragment into arthroconidia, which can be inhaled into the lungs. There it can cause primary pulmonary infection most commonly. Historically, as you can see in this map published by CDC, the endemic area for coccidioides is the southwestern part of the United States. The majority of people who are infected with coccidioides are asymptomatic. However, about 40% go on to develop primary pulmonary disease, and 5 to 10% develop severe or long-term lung problems. The incubation period for coccidioidomycosis is about 1 to 3 weeks in general. However, latent or reactivation infection can occur months to years later, especially in immunosuppressed persons. Follow-up molecular clock analysis using additional isolates from Washington State suggests importation to our state hundreds to thousands of years ago, so a relatively recent introduction, and this most likely occurred as a single or a small number of introductions from California. Climate suitability modeling, considering temperature and precipitation, successfully identified the area of Washington where we have identified cases. This is shown in the map with the pink coloring at the top. You can compare that to the map of the CDC endemic areas shown at the bottom. Future projections using climate data were used to estimate future habitat suitability for coccidioides, including in Washington state. You can see in the lower map that by 2095, this model projects expansion of the suitable climate of coccidioides to most of eastern Washington. Given this concern for a changing endemic area within our state, we used the findings from our initial genomic epidemiologic investigation to develop a new surveillance system for endemic coccidioidomycosis in Washington. Beginning in 2014, we started requesting case reporting from healthcare providers as well as laboratories, and we began sending these isolates to CDC for whole genome sequencing. We use the genomic data that's produced in addition to epidemiologic data about a patient's travel history or possible exposure events to differentiate locally acquired cases from travel-associated cases in order to maintain awareness of our endemic areas here in Washington state and to inform our prevention messaging. However, currently we do not have a database in use for managing or storing this data. The second example case study I'm going to walk through is a case of zoonotic tuberculosis. During August to September of 2019, the Washington State Department of Health received 10 positive IGRA test results reported among persons who worked at a zoo in Washington state. Subsequently, 2 Asian elephants housed at that zoo were culture positive for Mycobacterium tuberculosis. In conjunction with the local health department, Department of Health, and CDC, the zoo implemented a respirator protection program, advanced cleaning methods, ventilation improvement, as well as human and animal testing and treatment. Results of whole genome sequencing are shown here in red for the 2 elephants at the zoo. These isolates clustered closely together on a phylogenetic tree of other animal isolates and did not appear closely related to others. We did not identify any human cases that closely clustered with them, and so we were unable to identify a source of infection for these elephants. However, human and animal TB isolates are currently maintained in different systems, so this inhibits our ability to cross-reference across systems, and cluster detection between humans and animals is not automatically performed. It's important that we maintain ongoing human surveillance for tuberculosis that could potentially be related to these elephant cases. TB-GIMS, or the Tuberculosis Genotyping Information Management System, is the current database system that's used to maintain patient-level TB genotyping and epidemiologic data. CDC does provide support in this system for cluster identification for tuberculosis. However, as mentioned, it's currently limited to human data alone. The final set of genomic epidemiologic data systems that I'll review, and which cover some of our communicable disease history and genomic epidemiology, are those used for enteric disease cluster detection. There are 3 main systems in use that work synergistically together. The first is the PulseNet system, which is a national system for outbreak detection for foodborne and waterborne disease. This is a laboratory-based system by which laboratories upload genomic data that they produce to a central repository, and CDC monitors the system to identify clusters. In addition, NARMS, or the National System for Tracking Antimicrobial Resistance in Enteric Bacteria, is a system that's based on the submission of laboratory isolates from humans, from states that participate in retail meat sampling, as well as from animal specimens from slaughter and processing plants. And so this system is a bit more One Health in that it contains isolates from humans as well as animals and food systems. And then CEDRIC is a system that basically integrates the laboratory data that's available in PulseNet and NARMS, as well as epidemiologic data that's available from state and local health departments, and provides that data for use by local and state health departments, as well as by CDC. And so all of these systems I've discussed previously existed prior to the COVID-19 pandemic. But as you might have gathered, there was not one pathogen agnostic system and no easy way to link genomic data that's produced by multiple different laboratories to epidemiologic data that's gathered by local health jurisdictions, as well as the Department of Health. Early in the SARS-CoV-2 pandemic, academic laboratories were sequencing SARS-CoV-2 and publishing results. However, it wasn't until August of 2020 that Washington State Department of Health began looking into methods for genomic data integration into our public health system. By March of 2021, the Washington Department of Health had worked with partner laboratories to establish a sentinel surveillance system for our state. The map down below here shows the sources of different specimens submitted to sequencing laboratories. So those different sentinel laboratories would randomly select among their positive specimens and submit them for sequencing. In addition to increasing our state's capacity for the production of genomic data, we needed to work to implement systematic ways of linking that genomic data to our epidemiologic data and for storing that data over time. So this was a challenge because, again, there were multiple different laboratories producing this data. So we had academic labs performing sequencing, eventually our public health lab, as well as different commercial laboratories. So there were those commercial laboratories contracted by CDC, some contracted by Department of Health, as well as commercial laboratories not under contract, but producing sequencing data for their own use. And so we needed to figure out how to obtain identifiers from each of these laboratories in order to link to our epidemiologic data, such that we could produce reports of findings for the public. In doing so, we've been able to produce a weekly SARS-CoV-2 sequencing report. This is a PDF report that's generated in R Markdown. It's produced once per week and is freely available on our DOH data dashboard. You can find it today. And so within this report, each week we update these figures. And so we're able to look over time at an epidemiologic curve of the different variants circulating in Washington state. And so you can see the different ebbs and flows from the Delta wave to the various Omicron waves that we've seen. And so this data is lagged by about 10 days. And so it allows us to have a turnaround time for samples so that we can have some more accurate representation of data here, but you can go here to see what is currently circulating in Washington state. And we're also able to keep track of both the number of specimens sequenced and the number of sequences produced, as well as the percentage of reported cases in Washington state that have sequence data available for them. So as I mentioned previously, in March 2021 is when we first set up this sentinel surveillance program. And so you can see the increase in percentage of cases sequenced at that time. We've seen variability from 3% of cases sequenced during the first Omicron surge in January of 2020, all the way up to the high 20 percentiles of cases reported having sequencing data available. And so since implementation of this system in 2021, we've been able to step back and perform some evaluation of this system and understand what this has added and ways in which the system additionally may need to be improved. So on the left, we have prior to March 2021. So this is sequencing data that's available that was being produced either by academic or commercial laboratories, but really with no involvement of Department of Health. So this was before we had set up our sentinel surveillance system. As you can see, sequence data was not available representative of Washington state, and it was also largely available in Western Washington with large gaps in Eastern Washington. Below the map, what you can see there is a square for each county with the percent of cases sequenced over time. And so you can also see that there were some counties that might have had a batch of sequencing performed during one time period, but kind of gaps in the in different time periods during this pre-sentinel surveillance timeframe. And then on the right, you can see how this has changed since our implementation of sentinel surveillance in March of 2021. So we've really largely expanded coverage in Eastern Washington, which is important from an equity perspective, as well as we really do see different patterns of circulation of variants between Eastern and Western Washington, different timing of introductions. And so it was really important from a surveillance perspective to be able to capture this data more accurately and in a more representative way. And then you can also see in that same figure underneath the map, how not only have we increased our geographic coverage, we've been able to maintain those increases over time. So largely, we're not just seeing a spike in, say, collecting data from 1 month. We see data collected over time from each county and from different parts of the state. In addition to seeing improvements from a geographical and temporal perspective, we also saw large improvements in representativeness across many different demographic categories. So things like age and race, we saw improvements in representativeness. Some areas that have been a challenge for us are hospitalization status. So in order to capture cases who are hospitalized, you have to target different laboratories than just your standard outpatient laboratories. They may be labs that are actually associated with hospitals. So it has been a challenge for us to maintain representative sampling across kind of the disease severity spectrum. So that's one area we're still working for improvement. In addition to allowing us to maintain kind of situational awareness of variants circulating in Washington state and across different parts of our state, what this genomic epidemiologic surveillance system has allowed us to do is to start to explore some of the epidemiologic characteristics of variants. And so this particular figure is from a paper and project that we worked on with the Bedford lab and specifically Miguel Paredes, looking at the risk of hospitalization by variant. And so a project like this obviously requires that we sequence to be able to classify by variant, and also that we have linked epidemiologic data so that we can say something about the hospitalization status or disease severity. And so we were able to collaborate on this project and publish results with our findings around increased risk of hospitalization among beta, gamma, and delta variants. In addition, we're beginning to use our genomic and epidemiologic data for cluster detection and investigation. Cluster detection is a challenge because right now our genomic data takes an average of about 21 days from the time a specimen is collected until the time the data is linked and available for use by public health. And so 3 weeks is quite a lot of time. By then, we may have already heard about a cluster from an epidemiologic report. And so cluster detection is something we're still working on methods, both on timeliness improvement, but then also kind of validating our efforts for cluster detection from a phylogenetic perspective. And it has, however, enabled us to do retrospective investigation, which I think is largely how we've been able to apply methods around genomic and epi linked data. And so this is one example looking at long-term care facility clusters, what originally had appeared to be 2 different clusters of long-term care cases, one in blue and one in green. And we were able to determine that there was actually one staff member that was shared between the long-term care facilities, and that information did not come out on the preliminary epidemiologic investigation. But after looking at phylogenetic data, we were able to see that those 2 clusters were in fact linked and provide information to go back and repeat interviews and find epidemiologic data that supported the phylogenetic findings. And so we are able to use this data to look more in depth at clusters of interest, and will continue to do so over time. And so I hope I've shown from a couple of different perspectives, and I'm sure many of you are already aware and have been for a long time, that genomic data can add important information to public health investigations. So whether it's understanding the likely region of geographic exposure, like for coccidioides, it's understanding potential source and linkages between cases like for tuberculosis, or looking at more in depth at clusters and understanding epidemiology in different settings. It can be, really useful for prevention and mitigation methods. However, it's most useful when timely data and data linkage is available. And this has obviously been a challenge for us over time. And so when we think from a more broad perspective about genomic data surveillance systems and kind of what's required for the public health system going forward, I really think about it in terms of these 4 different critical components. So first, you need to be producing genomic data for that particular pathogen that you're interested in surveilling for. Second, you need to have capacity then for genomic data assembly and analysis. And this has been an area where public health, I think, has lagged a bit because we have not traditionally employed bioinformaticians. And it's an area where we are just, starting to build up our workforce capacity. We then need to have systems that enable linking genomic data and epidemiologic data, and systems that enable storing and retrieving that data over time. And so historically, as we've discussed so far, we've really approached this from an individual pathogen perspective. So foodborne and enteric disease have some really nice systems, as I described. Tuberculosis has their system for genomic and epi data linkage. And these systems at least do have support from CDC for ongoing monitoring and cluster detection. However, when it comes to other pathogens or emerging pathogens like SARS-CoV-2, at least in this pandemic, it was really left up to the states to figure out systems for linking our genomic and epidemiologic public health data. So we used largely GISAID and linked that to our disease reporting system. But it took a huge amount of effort and public health resources. And when we think about other pathogens, we really aren't set up at this point to be able to do the same thing over time. And so what we're doing at Department of Health in order to address this concern is that we've started our molecular epidemiology program. This started as a single staff person back in August of 2020 and has grown to now 6 staff people. It has undergone a lot of program development, and we've developed this mission to be a national leader in genomic epidemiology capacity building and a resource for public health partners, with a mission focusing on the integration of genomic and epidemiologic data across pathogens that we surveil for, the development of best practices in genomic data analysis and visualization, as well as interpretation and communication for public audiences. And so in regards to capacity building, what we're doing is supporting the prioritization of other pathogens for sequencing at our public health laboratory. And so we're really using our forward momentum from SARS-CoV-2 and from implementing sequencing here in Washington State to pivot to some of our other notifiable conditions. And so currently, even though I discussed those surveillance systems that we have in place for, say, coccidioidomycosis or tuberculosis, all of the sequencing that's performed for those organisms happens at CDC or at other laboratories. And so really focusing on bringing that capacity in-house, that will help us both with our genomic to epidemiologic linkage projects. It can also help us with prioritization of timeliness. So right now for coccidioidomycosis, it takes about 6 months for us to receive those results back. And so we really look at that as kind of like a long-term system of understanding endemic areas in Washington, but it doesn't allow us to perform like timely public health action as a result of individual cases. And so exploring how increasing timeliness might support public health investigation. We're also building capacity both within our state health department as well as local health departments for performing and conducting genomic epidemiology. So we're hiring this team of EPIs to support our other notifiable condition program areas. And then in addition, we're offering office hours to help with training and capacity building for local public health and tribal public health in order to support their use of genomic data as well. So our team is really working hard to learn from past and current surveillance approaches. And so a major part of this effort is conducting an assessment that crosses our various condition areas to ask those questions about how genomic data has been obtained and used and stored in the past, and best practices in those particular fields that are known to be out there, as well as preferred mechanisms for moving that forward here in Washington state. And so this is a pretty large assessment that's currently underway that will hopefully help inform how we move forward, not just for individual condition areas, but how we can identify kind of best practices across the condition areas. And then in addition, we're joining national conversations on systems development. So CDC has recognized, that there is not a national platform for the linkage of genomic and epidemiologic data, and has recently announced some grant funding for kind of pushing that priority forward and including states in that conversation. And so we are working with CDC to join that conversation to make sure our voice and our experience here in Washington state is heard to push these priorities forward. And so all of this work is supported in a huge part by the clinical and sequencing laboratories that make this possible through sharing data with public health. And then each of the investigations that I spoke about previously were supported by different teams, both local health and Washington State Department of Health and CDC. And then a huge thank you to our current molecular epi team, which includes Stephanie Lunn, Marcella Torres, Lauren Frisbee, Kathleen Connery, and Rebecca Tomier. Our public health lab has gone through enormous efforts to bring sequencing on board for SARS-CoV-2, and again is now pivoting to look at other conditions. And so huge efforts on that front, as well as really great efforts from our data integration and quality assurance team as well. So I am available for questions. We also have this WGS Epi, which is the group inbox for our molecular epi team, if you all have questions in the future. But for now, I'm happy to open it up and take questions on the call.
Great. Thank you so much, Hannah. That was a great overview of the development of the program. There are a lot of questions in the chat. I think what I'll do is go ahead and just read them out to you and have you answer them. And then Trevor, there are some for you too, but it sounds like most of the ones that were asked of you have been addressed, but I'll also go ahead and give you some space to answer those. So there's a question from Lauren Rhoda. And the question is for you, Hannah. It's do you have records of prior infection or vaccination for the individuals for whom samples are sequenced? And is it possible to say how much the decrease in hospitalization with successive variants comes from immunity versus a change in the variant?
Okay, so that's particularly for SARS-CoV-2. And it sounds like that question was around the hospitalization study. And so, yeah, we did have epidemiologic data linked to our genomic data for that study. And so we were able to control for confounding and we did have information on vaccination status available as well, yes. In regards to previous infection, so we excluded cases that were known to have a previous infection from that analysis. However, it is possible, Of course, we know that not all infections are symptomatic, and not everyone necessarily is tested for every infection. And so we didn't have a way of controlling for unknown infections. So infections where we didn't have test results available or that weren't reported to Department of Health. But for those that were known and for vaccination status, we were able to control for those.
Great. There's one more question from David Gordon about the reporting frequency, and that Washington State has decreased reporting from 5 days a week to 3 days a week, but often there's a holiday on Monday. And the request is, could the reporting be provided at minimum 3 days a week? And if not on Monday, then could it be provided on Tuesday? I'm not sure if that's something that you would be
wondering, is that just like SARS-CoV-2 cases in general? I'm wondering if we could have clarification, because our sequencing report is a once-a-week report, so it sounds like that's referring to case reporting in general. And if that's the case, I have no control over that, but good feedback.
I believe it is related to cases in general. And then Trevor, you have a question for Hanna. Do you want to go ahead and ask it?
Sure. Yeah, I'm of course wondering about BA5 and like whether we can say something from Washington State from early data. Is there like enough data for severity estimates and whether that differs from previous Omicron, and like also symptomology? Do we know if there's any differences there among people? Yeah, perhaps still too early days, but I'm wondering if anything can be said at this point.
It's a good question, and I'm going to start with addressing the question about symptomology, because I think our ability to capture epidemiologic data around SARS-CoV-2 cases has really changed since the Omicron wave. So prior to Omicron, we were still attempting to contact and interview all SARS-CoV-2 cases that were reported to Department of Health. And since Omicron, the number of cases has far outweighed our capacity. And in addition, I think there's just been a movement toward understanding that we're no longer in a place where contact tracing is really supporting public health goals. And so we no longer have symptom data available for the majority of cases. So I think that that's a big limiting factor there. We do still have hospitalization data available. We're able to get that through other systems. And in regards to numbers for BA5 cases, I'm wondering, are other participants able to unmute themselves? I would actually ask someone on my team. If not, we can get back to you on that, Trevor, but I haven't looked at numbers of BA5 recently.
I see. I think anyone can just like talk if they want to.
Stephanie or Marcella, I'll ask if you're on. Are you able to speak to current numbers?
Hi, this is Stephanie. Can you hear me?
Mm-hmm.
Perfect. Yes, we are looking into hospitalization for BA5. It seems that we don't have a lot of data just yet, but we're working on getting a more accurate count.
And Trevor, we can probably pull the symptomology data for you from the University of Washington testing program, where we do have symptoms. It didn't appear to me that that might be a data need, but we can get that information over to you.
Cool. Yeah, it's funny where like I guess we had pretty early results for Omicron severity differences from like Imperial College and so forth. But like with each of the Omicron subvariants, things have been generally slower for that kind of data too, for those sorts of estimates to come in.
Do you think it's because there have been so many overlapping waves that it's difficult to parse out?
Maybe, and I don't know if I think is all like confounded by like if people were to like be concerned about this thing and call it a variant of concern and like just like treat it as such, it might be different versus it's all Omicron and we don't, it's all Omicron. I don't know if that's really fair, however.
Great. Hanna, can I ask you two questions? One is, Is there, can you explain to me the relationship between the Washington State Department of Health and Public Health Seattle King County, and how the data is integrated across both groups, and how you harmonize in terms of case counts and transfer of data, and whether or not that could be a way for vaccine data to be better integrated? And then my other question for you is about what the efforts are on the state level for collection of home antigen test data. And if that's something where a system is being developed. I don't know if you know, but at University of Washington, we're using a QR system for people to upload their antigen test results, and linking that with a request to then get a molecular test so we can do genomic surveillance. And I'm wondering if that's something that Washington State has explored.
Yeah, so I'll start with the question about King County first. So I guess just basics on the public health system in Washington. So in the US, there are two different types of public health systems in states. One is centralized and one is home rule. We are a home rule state, which means that our local health jurisdictions have the public health authority and own the data. So we have 35 local health jurisdictions in Washington State, of which Public Health Seattle and King County is one. And they are all required to report to Washington State Department of Health. And so in all cases, Public Health Seattle King County is truly the data owner, and then they report up through a shared data system to Department of Health. In regards to genomic data, Department of Health has acted as the curator, because we are the only ones really who have the ability to link the data back to epidemiologic records. And so then we share that information back through this shared data system to Public Health Seattle and King County. And so I'm not aware of additional data sources for vaccination that King County has that Washington State Department of Health does not have. So we all have access to IAS data, which is our immunization information system in Washington State, and that is linked to our epidemiologic data set. And then in regards to antigen testing, so I know right now, the way that that's done is that people are encouraged to call into a phone line and report results. My understanding is that's very incomplete capture, and I'm not certain. I could look into whether we are pursuing a QR system, but I think in general, and I'll speak kind of in general from the public health system, really across the US as well as in Washington State, I think we've been moving away from trying to count cases. So from counting every case, if that makes sense, and trying to understand more trends and kind of the big picture, because I think at this point in the pandemic, so many people aren't testing. There's also really large differences in case capture by geographic area. So you have people testing at different rates in different parts of the state. And then, of course, the use of home antigen tests as well, making it difficult to capture. And so instead of trying to count every case, like for influenza, we make no attempt to count every case. We use syndromic surveillance data as well as some sentinel surveillance data, and kind of more efficient systems to try to look at that. And so that's kind of the general approach that it seems like we'll be moving to towards SARS-CoV-2 in the future. Even though we still publish case count data, we know that it's fewer than 20% of cases that are actually occurring out there in the population. And so I think at this point, quite frankly, it's misleading to be publishing case counts, because it's not what's happening out there. We really need case estimates. And so yeah, all that to say, I'm not aware of efforts to pursue that at this time, but I may be incorrect. There may be a team working on that at Department of Health.
Do you think there's opportunity for kind of more like randomized surveys here, where you could like call people and ask, have you had a positive antigen test in the last month or whatever it is, or some other I know we've talked about this before, where like the UK infection survey of mailing random swabs to people has been extraordinarily powerful. And like, again, whether less data, but randomized sort of data might be able to get at these prevalence incidents in a way that we're just not getting because we can't capture every case.
Yeah, there's definitely been a lot of interest in that. And I think my personal perspective is also that would be, the most efficient and effective way forward if you're just looking for what data will give you the most bang for the buck. However, it is expensive, and we currently don't have funding streams for that type of work. However, to my knowledge, we are looking into what funding streams could possibly be made available to do something exactly similar, mailing swabs and that type of thing, because I think there is a lot of recognition at this point in the pandemic that it would be more useful to have a random subset of data.
Thank you.
Thanks, Hannah. One more question. Does the move away from Judy, does the move away from case counts mean any change in the application of genomic surveillance to wastewater in Washington?
Yeah, so I think, so we are still maintaining our Sentinel surveillance sites for like individual case monitoring for genomic data for the foreseeable future. However, in addition, we are very interested in expanding our system to include data coming out of wastewater. So right now there is a lot of work going on to identify different facilities and catchment areas that could participate in that type of surveillance in Washington state. There is already surveillance going on in several different parts of the state, and we are already sequencing from some of those samples, but we're basically just in the early stages of figuring out how to integrate some of that reporting into our existing case-based data streams. But yes, I also think that is kind of the way of the future, looking more at a population level and less at, counting individual cases. There are some real challenges with sequencing from wastewater, or even from like enumerating estimated like positives from wastewater, because it can be affected by weather and by the number of people, in a catchment area and that type of thing. And so there are a lot of challenges to work through and some methodologies that need to be developed, but we're certainly moving in that direction as well.
Okay, great. Thank you. Trevor, there were a couple questions for you. I don't know if you wanted to elaborate more about RT and R naught, and then also about the RT for BA 2.75 specifically.
Yeah, very briefly. So R naught should be given the current behavior and so forth in the population. And so R naught can actually change. It depends upon the pathogen and its human behavior of the kind of population that's in. So R naught for original SARS-CoV-2 was maybe 3, maybe 3.5, something pretty significant. That's ratcheted up over time as the pathogen has become more intrinsically transmissible. I think at this point it's probably 8-ish, which is really, really quite large. And then, but we also have antigenic drift that kind of decays immunity. And so RT is going to be what is the number of secondary infections that are caused at this point in time, and in what I was showing by this variant. And that we can estimate from, even if the cases aren't capturing every infection, you're looking at like, are the cases increasing or the cases decreasing? And so as long as like your case detection rate isn't varying too rapidly through time, you can still estimate RT just from changes in case counts. And there, by looking at, so for BA.2.75 in particular, you can break down cases in India based upon variant by looking at frequency in the sequencing database. And we can see that frequency of BA.2.75 is shooting up really, really quickly. And so we'd estimate initially that RT T for BA.2.75 is really big, but then we're not seeing yet those kind of overall cases increase. And so that's why kind of RT dropped to 1.2 over the past 4 weeks in India, at least our estimation for this. And so it's going to be, again, we can look in the next week or so to see maybe what's going on. Okay, yeah, thank you.
Great. I think we have reached the end of our questions. So thank you to Hanna and to Trevor for speaking today. This has been a really great talk, and it's been really valuable for us to hear the perspective from Washington State. So, and you've really played such an important part in the response to this pandemic. So thank you for all your work. As a reminder, we are taking a break in August, and then we'll be coming back in September. And our speaker will be Pavitra Roy Chowdhury, who is at the University of Washington in the clinical virology lab, and we'll be giving a perspective on genomic epidemiology from the perspective of the University of Washington. So thank you all. The recording will be up, and we'll see you in September. So happy summer, everyone.